Hey guys, Mr. Hyatt here. In this walkthrough we're talking about ribosomal structure and function, which is sometimes called translation. So ribosomes are made of two subunits. It's kind of like saying there are two parts that come together. About 60% of that, or the large subunit, is made of RNA. Sorry, ribosomal RNA, that's that specific kind of RNA. And then the smaller subunit is 40%, and that's made of protein. Notice that it says rRNA is the most abundant type of RNA in a cell. That shouldn't surprise you if you think about the fact that mRNA is messenger RNA. It's only used for a short time and then it's destroyed. tRNA is so unique and specific that it would be impractical for us to have tons of it around all the time. So we do have lots of ribosomes around all the time. So it should make somewhat sense that rRNA would be the most abundant type of RNA in a cell. Here we got a couple of pictures. You can see some proteins and the rRNA. This is kind of notice, sorry, this is the large subunit. So this is the bigger chunk of it. We've got another picture that's going to show the two subunits together. So notice that the large subunit is even an interaction between proteins and rRNA. Here's both subunits together. You can see the proteins in this picture are labeled purple. Where they were silver or gold here, sorry. So you can see it's a complex structure. We're going to simplify it down and use some models to try to understand what's going on here, but it's a pretty complex chemical process that we're going to simplify down, and I think will make a lot of sense by the time we're done. Excuse me. So the large subunit has three sites. So we got a P site, an A site, and an E site. That the names, the P site and the A site, obviously deal with the which type of tRNA we're talking about. For me, it helped to remember arrival site, processing site, exit. Maybe that's helpful, maybe it's not. But that's essentially what's going to happen. That's how we're going to build a protein off of a nucleic acid strand. We're going to work our way down one at a time. So I'm getting ahead of myself. We got three steps in translation. So the first couple of slides were about the ribosomal structure. Here's the ribosomal function. Translation, we're talking about translating languages. So we're going language of the nucleus to language of the cell. So we're really changing drastically here. This is going English to Spanish. We're really changing languages. We got three steps. You got to start, you got to build, and then you got to stop. There's a video link. I've linked this into Canvas. This is a really good animation that I've used for years in teaching translation. It's got a lot of the specifics. I don't need to spend time going through it. It's there. Use that resource. Here's basically the process. Initiation is going to involve a couple of coordinated steps. We're going to have the large and the small subunits are going to come together. So notice that the small subunit latches on to our mRNA transcript that's coming from the nucleus, and excuse me, it's going to locate the methionine tRNA. There's our anticodon. There's our AUG that's signaling for MET. Excuse me. This is all part of initiation. Initiation kind of finishes when the large subunit assembles with the smaller subunit, and now we've got our complex. The tRNA that's holding the methionine is in the processing site right now. The next amino acid will arrive at the A site, and then we'll be talking about elongation. Okay, So, sorry. Initiation is going to require things called initiation factors that are going to promote it. I think that video talks a little bit about those. And then GTP is used as an energy source. I left that in here because I feel like I need to point out every time that we use GTP because it's so infrequent. Okay, so we talked initiation. Now elongation, we're building our peptide. The peptide's getting longer. Okay, so we've got recognition. We've got this anti-codon that's going to match our codon of the A site. The tRNA is going to bump into the A site, and that's called recognition. Essentially, we're just talking about matching up the anti-codon and the codon. The peptide bond is formed between the new amino acid and the existing polypeptide or amino acid, if we're talking about the second one, that's in the P site. So that's why, to me, I called that the processing site. This bond is accomplished by an rRNA, ribosomal RNA, acting as a ribozyme. We talked about ribozymes in class, so that should sound familiar. So we add GDP, we get a tRNA in the A site, and we've got a polypeptide sticking off the tRNA on the P site. Excuse me. Okay, so now we get our bond formed across between these amino acids. When that bond happens, the connection to the tRNA in the P site is broken, and our polypeptide chain just gets a little bit longer. Next step, excuse me, the ribosome is going to work its way down. It's going to pull the mRNA through. This tRNA is going to be kicked out. It's going to exit, and we're going to have a second or a third or a fourth or a fifth, or the next tRNA is going to arrive at the A site, and we're just going to keep working our way through. Sorry, I stumbled through that. So elongation essentially takes 60 milliseconds for each amino acid that's added. This is a pretty quick process, which is good because our proteins, as you saw, if you just look at the ribosome structure, our proteins are complex. So we got to be pretty quick at doing this, and we are. Termination is going to be stopping. We're going to come across a stop codon. We're going to get a release factor binding into the A site instead of a tRNA. So that's the idea behind that is similar to what we saw in DNA replication. The implementation is a little different. So water gets added instead of an amino acid because of this release factor. So that's going to free the polypeptide. The ribosome is going to split up, and things are basically just going to split. And that's what we're seeing here. Notice our release factor has an anti-codon, structurally somewhat similar to the tRNA, but doesn't have an attached amino acid. That's going to allow for hydrolysis. It's going to cause the two subunits to break up. The mRNA is going to float away. The protein is going to float away. And the release factor is going to float away, and they'll all be recycled. That's kind of the basics. But we usually don't need one copy of a protein. If we need a protein, we need 30 of them. So we need to make multiple copies at a time. Well, one way we can do that is with these things called polyribosomes. They look kind of like beads on a string. So each of these ribosomes is building a protein, building a polypeptide. So we've got 1, 2, 3, 4, potentially 5 ribosomes all building proteins off the same mRNA strand. So that's one way that we can amplify our production. Here's an example of that, again, beads on a string kind of thing. In a prokaryote, they have a closed plasmid type of a looped DNA. So it just has to happen a little bit differently, of course. So you see, excuse me, I'm sorry, that these things are kind of branching off the DNA versus in the eukaryotes, they were kind of wound through. There is something called the signal hypothesis that's pretty interesting. Essentially, it says there's something on the mRNA that causes ribosomes to attach to the rough ER. Otherwise, ribosomes are free-floating, which kind of makes sense because these things don't assemble until they're recognized by an mRNA anyway. So maybe part of initiation is this signal receptor protein connecting to the rough ER. If it's close enough. If this happens somewhere out in the cytosol, chemically that's not gonna be able to happen. So the effect is that proteins are made directly into the ER, which is great because the ER is how we ship things around. If we're going to use it in the cell, we're going to use the protein in the cell, we're going to use the ER to transport it to the Golgi, and it's going to be targeted. If we're going to excrete it, again, we're going to use the ER to ship it to the Golgi, and it's going to be sent out that way. So again, it lets us ship more accurately. The clue, which is like a primer, like we would have called a primer in our last chunk of material, chapter 16, it's processed out. It's like an exon. So it gets pulled out. I'm sorry, like an intron. It gets pulled out and gets processed out. So the clue isn't expressed, doesn't become a part of the protein. So that's ribosomal structure and translation. Hope this stuff's starting to make sense.